Have you ever actually stopped to think about the absolute limits of human biology? Because no matter how closely we squint, there is an entire bustling universe happening right in front of us, on our skin, in our water, on the literal leaves of plants, and it's entirely invisible to the naked eye. So, how exactly do we explore a world we can't even see? Well, we have to build a bridge made of glass, light, and serious mechanical precision. Welcome to this explainer. Today, we're focusing squarely on a singular, literally world-altering invention, the compound microscope. More specifically, we're going to unpack exactly how these microscopes use lenses to make the invisible, well, visible. It's this fascinating bit of mechanical magic. And by the end of this, you're going to understand exactly how the whole shebang works. To get us there, here is our quick roadmap. First, we'll cover the invisible world. Then, the power of two lenses. After that, we'll follow the journey of light, calculate magnification, go over proper use and care, and finish up with a world-changing invention. Okay, let's jump right into section 1, the invisible world, defining the tool. Let's start with the name itself. The linguistic roots of this instrument are elegantly simple, right? You've got the Greek mikros, meaning small, and skopin, meaning to look at. But its true function, that is profound. It doesn't just magnify, it entirely redefines our understanding of biology by letting us actually observe the literal building blocks of tissues and cellular structures. It is the key to an otherwise completely hidden biological kingdom. Moving on to section 2, the power of two lenses, a tag team approach. So the word compound is really the secret ingredient here. It means we aren't just relying on one powerful magnifying glass. Instead, we're using multiple lens systems working together in a sort of relay race. They take a microscopic image, catch it, and literally blow it up to a scale our eyes can actually comprehend. This teamwork is what sets this instrument completely apart from, say, a simple jeweler's loupe or a basic reading glass. And that brings us to the two main players in this tag team approach. Up on one side, you've got the eyepiece, which is also known as the ocular lens. This is the lens at the very top, the one you actually put your eye right up to. Think of this as your reliable anchor. It usually provides a standard 10 times magnification all on its own. Then, on the other side of the equation, we have the objective lenses, located way down close to the specimen itself. These are the dynamic variables doing the heavy lifting of the primary magnification. And a physical body tube is what connects that top anchor eyepiece down to those lower objective lenses. Let's break down that dynamic lower half, which I like to call the objective arsenal. These lenses sit on this rotating disc, a revolving nosepiece, which lets you just effortlessly swap them out. You always start with the scanning objective. That's a 4 times magnification, and it's used simply to locate your specimen. Next up is the low-power objective at 10 times, allowing you to observe more details. Then things get serious. You've got the high-power objective at 40 times to observe the really small details. And finally, some microscopes actually feature an oil immersion objective at a massive 100 times magnification. Now get this, it's super fascinating. At 100 times magnification, light actually scatters too much in the open air, so scientists have to use a drop of special oil to bridge the literal gap between the slide and the lens. This keeps the light totally focused and delivers maximum detail. No way around it. All right, section 3. The journey of light, illuminating the unseen. You see, all this magnification is made possible by a perfectly straight path of light. Illumination literally shoots up from the base, it gets concentrated by a lens beneath the stage called a condenser, and then blasts straight through your specimen. The objective lens catches it, sending it right up the body tube where the eyepiece magnifies it one last time before it hits your retina. And here is absolutely the best part. Because of how light bends through these curved glass lenses, the final image you see is completely inverted. Seriously, if a single-celled organism swims up and to the right on the physical slide, when you look through the eyepiece, it's going to look like it's swimming down and to the left. Kind of wild, right? Which leads us to section 4, calculating magnification, doing the math. Now, don't worry, the math here is incredibly straightforward. Because we're using a compound system, the total magnification is calculated by simply multiplying the magnification of the eyepiece by the magnification of whichever objective lens you currently have clicked into place. It's just a literal multiplication of our tag team of lenses working together. So, to put that into perspective, if you pair a standard 10 times eyepiece with a 40 times high-power objective lens, you just multiply them. 10 times 40 equals a massive 400x total magnification. The specimen appears 400 times larger than reality, all thanks to that compound optical system. Next up, Section 5, Proper Use and Care, Wielding the Power Safely. Now, wielding this kind of optical power absolutely requires discipline to protect the glass. You always start by clicking the lowest power objective, that's the 4x scanning lens, into position to map your terrain. You place your glass slide on the flat platform, called the stage, and you lock it firmly in place with the stage clips. When you bring the stage up with the coarse adjustment knob, you have to watch from the side, not the eyepiece. Why? Because the absolute golden rule of microscopy is to never, ever let the objective lens crash into the glass slide. Only once the stage is set do you actually look through the eyepiece and bring the image into initial focus. Okay, so once you have your target, you refine the image, you dial in the light with the condenser to get the perfect contrast, you center the sample perfectly in your field of view, and then you tease out the absolute sharpest details with the smaller, fine focus knob. Only when it is crystal clear do you carefully click to the next level of magnification, making minor adjustments as you go. And when the exploration is totally over, you lower the stage, click back to the lowest power, and very carefully remove the slide. These precision lenses demand a lot of respect, and protecting them is absolutely paramount. You have to carry the microscope like a precious artifact, one hand firmly grasping the arm, the other supporting the heavy base. And you never let your fingers touch the glass. Natural skin oils will actually degrade and permanently etch those expensive optics. You should only use specialized lens paper if a cleaning is absolutely required. When the exploration is over, you cover it right up to keep the dust away. Basically, treat the lenses well, and they will reveal the world to you. Finally, Section 6, a world-changing invention, the impact of lenses. This gradual stacking of curved glass literally rewrote human knowledge. Way back in the 1590s, the Janssen family was experimenting with basic compound tubes, but it was in 1665, when Robert Hooke used improved lenses to look at cork, that he actually coined the term cell. Just a decade later, Antoine van Leeuwenhoek peered at pond water and was absolutely stunned to find living, swimming microorganisms. He affectionately called them animalcules, which is just a great word. Over the next century and a half, scientists like Schleiden, Schwann, and Virchow used these exact compound principles to prove that all plant and animal life is cellular and that cells only come from other living cells. This single arrangement of lenses birthed modern biology and medicine as we know it today. Which leaves us with this final thought. The invention of the compound microscope proved that our physical limitations really don't have to be the end of our exploration. By simply bending light through layered pieces of glass, we unlocked the foundational building blocks of life itself. So, if simple lenses could unlock the cell, what invisible worlds are just waiting for us to build the right tool to uncover them next?